Well, good morning, ladies and gentlemen, and welcome to the Boffin Slug-Resistant webinar. My name's Tom Allen-Stevens. I lead the British On-Farm Innovation Network, and what you'll be witnessing this morning is history being made. But before I introduce today's main event, I'd just like to give a bit of a preview of some of the other exciting projects that we have coming up this year. And now I'm just going to share my screen. Bear with me. So for those of you who joined last time, you'll know that we have some fascinating projects coming along this year. We have a webinar to update you on these every third Thursday of the month. Now, so that there's a real opportunity to get involved in these projects, and for many of them, that will also include being paid to carry out on-farm trials. And that's not just trials for your own benefit, they're to advance science and to accelerate productivity for all UK farmers. So first of all, we've been talking with RAGT about insecticide-free wheat. And now this is only really possible thanks to new genetics coming forward, and particularly BYDV resistance. And they're keen to work with you to understand how we can really make the most of these genetics. And of course, the new £45 a hectare payment under SFI for not applying insecticides. So the date of the webinar is the 20th of April. We've got a lineup of speakers to explore what's possible with insecticide-free wheat. And looking to discuss what support you need if that's the journey you're on. So look out for the invite that'll come out shortly for that webinar. And then there's harvest weed seed control. So this looks at assessing the amount of grass weed seed that's still standing at harvest and available to mills such as the Reddycop Seed Control Unit that's fitted to the back of the combine. Now we've secured funding for a second year of trials, and we'll be looking for seed scouts to get involved in some really groundbreaking research. So more on this in the webinar is planned for May. And then we have a very ambitious and exciting project due to get underway shortly. And this is a large industry consortium led by PGRO, look out for more on this project when details are announced. And we're planning a kickoff webinar in June. So, now, first, let's move on to today's main event. So in a moment, I'm going to pass over to Keith Walters, who will deliver the first year results of the trials. Then Simon Griffiths will tell us about the feeding trials that have just got underway at John Innes Centre and the genetics of the Watkins lines that we're studying in this trial. Then we'll bring together two of the slug sleuths. So Richard Cross and Graham Potter will share their experiences of carrying out the trials. We'll then open up for questions. Now, there's a Q&A and a chat button that you'll see at the bottom of your screen. Please put any questions that you have in there. And towards the end, I'll bring you into the discussion so that you can put your questions direct to the panelists. And that's if I can work out the technology. And please do get a discussion going. Your thoughts and ideas really do shape the work that we're doing in these trials. So just to recap, the project got underway last June and revolves around Watkins 788. Now, this is a landrace variety that was identified in feeding trials at John Innes Centre as one that was consistently spurned by slugs. Now, only feeding trials can actually confirm if there is a slug-resistant trait. Only field trials will actually confirm if there is a slug-resistant trait that could help farmers move away from a reliance on chemical control measures and the potential impact of those chemicals. And that's what today is about. We have 10 slug sleuths conducting those very trials on the first ever plots of Watkins 788 to be grown on British farms. Now, what's important to note here is that this potential trait may never have gone anywhere had it not been for you, the Boffin members, who asked to be involved in an on-farm trial. So, how's it going? Well, I'll just highlight a couple of important developments. Firstly, I'm delighted to say that the Environment Agency has very kindly come forward and supported the trial. And that's helped us, allowed us to conduct more feeding trials at John Innes Centre, which are underway at the moment. And you may have seen that we are appealing for slug scouts to trap and send in slugs for the trials. So please do get involved, and I'll say a little bit more about this later. But look, that's enough from me. Time to hear from the real expert, who is Keith Walters. So Keith, are you there? Can you join us, please?
Am indeed, and I
Fantastic.
know I'm
Let
going
me
to be
just
wrestling to see if I can put on my slides. And sooner or later, a bit of luck and a fair wind.
Yeah, we're all there, I think.
Yeah, there we go. That's what I was waiting to do, to get some responsiveness. Okay, well, thanks for that, Tom. And thanks for the background. That saves me a complete slide, as you've seen, which has dropped off. We are now 1 year into this trial program. We've had the first initial trials completed, and we had two objectives which we set ourselves for that trial. One was to investigate the potential for on-farm research trial delivery against a background of restricted availability of seed. And secondly, to develop and test a trial design investigating 2 winter wheat lines with potential for slug resistance. There was, of course, a constraint here. There was very limited amount of seed. This is a first year of a trial series, and this restricted within real trial replication and total number of trial sites. And therefore, we needed to develop, as well as the field design, the statistical analysis design, which gave meaningful results despite these very severe constraints. The results we came up with will be familiar to many of you. This is the trial design. We compared in most of the trials, farm standard with the Watkins 788 seed. And in some of them, we also had Blackman seed. The reason we only had 3 trials with Blackman seed was simply that we didn't have enough seed to set up anymore. There were several critical aspects to this trial design, which were make or break in terms of them working. I won't go into all of them now, they're rather technical, but just as an example, we stipulated that the traps we were using, the surface refuge traps, to assess the number of slugs in each of these trial plots had to be a minimum of 5 meters apart. This wasn't just taken off the top of our heads. We'd done some extensive work beforehand, where we'd actually surgically implanted the tags which vets put in your cats and dogs so they can be identified if they get lost. These tags allowed us then to follow the slugs, whether they were living above or below the soil surface for extended periods of time in the field. And this example, for example, shows their movement from an initial release point, and the scale is 2 meters. These are 2 meter squares. What we found with this was that whichever of these slugs we looked at, they were within, after 5 weeks from their initial release, found just approximately 1 to 2 meters away from their initial release point. Now, over that 5 weeks, of course, some of them had moved up to 4 meters apart, but had always come back homed in towards their normal release position. And therefore, we knew that if we had the traps 5 meters apart, then they weren't interfering with each other. They were actually looking at different slug populations within these plots, which was critical for the statistical analysis. So that's what we did. We set ourselves a number of aims in this year 1 to test how well we could do these trials on farmers' field. And the aims are in yellow here, the outcome is in white. The first one was a simple one, run 10 sites. And indeed, we got all 10 sites established. All were rigorously set up and measured out really well. 7 of those sites had Watkins seed compared to the farm standard seeds, what the farmer would normally grow only. And 3 of them had, in addition, Blackman seed. Oops, lost it. We asked the farmers to visit the area twice a week or less frequently when tillering was well underway. And this also was followed rigorously. We had between 8 and up to a maximum of 13 assessment visits per site amongst all 10 of those. It worked well. Slug activity was measured using some surface refuge traps, and we got complete slug counts at 7 sites. And I'm sorry, that's a mistake there. All 3 with Blackman's seed also had that we got complete slug counts. One thing I would point out here is this was done exceptionally well. You can imagine that the site, for example, where there were 13 assessment visits, that one had Blackman's seed as well. So that was 6 sites, 2 traps per site. There were hundreds of traps, which we asked people to count the slugs under and record them. Out of all those, I can tell you that there was only one single missing piece of data, one trap, which wasn't actually counted. And that is well up to the standards you would expect from an experienced technician. We asked people to photograph the crop growth to ensure there are some stipulations that it was taken from the same angle and distance above the crop was maintained from for the point you took the photograph. And again, here we got some good results. We had 3 full sets where every trap was photographed on every assessment. We had a 4th site at which just 1 single photograph was missing, and 3 sites where there were partial photographs needed in year 2 to analyze. This enabled us to look at emergent state and also using pixel analysis to start testing the procedure for future years in order to look at crop coverage at each assessment visit. More on that later. We'd asked people also to score the growth of each plot, and we had 7 sites with these complete crop condition assessments. Again, this was good. I would stress that the reason for this, and for some of the other figures above, is that at 3 sites, there were absolutely no slugs whatsoever. And so they were quite understandably abandoned to save time.
Keith, sorry, can I just ask, you've got it on split screen at the moment. Is there any way you can get it to full screen?
I'm looking at full screen myself, actually, at the moment. I'm not sure what's happening there.
I don't know whether it's something to do with how Zoom is representing it. There seems to be a thing in the bottom below your screen. If you move your mouse to the bottom, there's some icons along the bottom. Can you see those at all?
Yeah. Okay.
No. If you start again, start your share again.
Okay. Does that help?
And now, if you start it again, hopefully that'll.
It's sharing now, apparently.
Yep, it's sharing now. So if you start your slideshow again, apologies for this.
Yeah, this is we're slow today on responsiveness here, so this might take some time.
Are you on full screen, Keith, where you are?
That's what we're waiting for. Yeah.
Oh, you're waiting.
The response time is slow here.
Great. That's fantastic. That's much better. Thank you very much.
Okay. Right. Okay. Sorry. I'm not sure what happened there. I've got exactly the same view as I had beforehand on mine, but at least it's working for you. Okay. The final thing which we have asked people to do was to harvest the plot separately and record the yield of each plot. Of course, it's too early to report here. So the outcome of this, what you can see from this brief summary, is that the detail of the trial requirements was delivered virtually flawlessly. This was an excellent set of data, and it really, really is, and I'm not just saying this, up to the publishable standard which one would expect from professional technical support. Moving on to the second objective, which was to trial design testing. This worked well as well. Using the data that was collected on slug numbers, what we did was to compare the farm standard plots with the Watkins wheat either untreated or treated with pellets, and the Blackman wheat. Here, initially on this slide, we're looking at an initial analysis to see if there was a trend in the direction of differences between treatments. In other words, was there always more slugs in the farm standard with, in this column, the Watkins when compared to the Watkins 788 untreated plot, and similarly with the Blackmans. And we analysed the untreated and the treated comparisons separately. Now, just to explain this, these blank columns here are simply where the trials weren't run because we didn't have enough seeds. So those are the results we had for Blackman. Oops, keep tapping the, there we go. And here, where there is a dash, it is where we didn't have sufficient degrees of freedom. That's a statistical term looking at the strength of comparisons which we can get, and we didn't have sufficient in order to make a worthwhile test. Reason for that was that universally through this, slug numbers were very, very low. So, for example, in Becky's site here, there were absolutely no slugs counted. There wasn't a single slug found throughout the whole trial in any of the plots. So clearly we couldn't make a worthwhile comparison. However, as slug numbers, mean slug numbers increased, what we found was we began to get in increasingly to a position where we could actually look at statistical comparisons and make meaningful comparisons. But again, because the numbers were so slow, most of these showed no significant difference. There wasn't a consistent difference between the number of slugs in the farm trials and those in the farm standard plots and those in the slug-resistant wheat plots. Finally, when we got to the very, very highest number of slugs we had, we started getting a difference, and this was quite a strong, significant difference there. Now, what's happening here? Is the trial going wrong? No, it's not. What we found in other work is that using the traps which we employed in this design, which are known to be the best ones for this kind of work, you cannot start getting significant differences between the traps at slug populations much below an average of 1.2 slugs per trap. And as you can see, we are actually, in statistical terms, well below that level. But what we do see here is as the populations get larger and larger and get closer and closer to the 1.2 per trap, we start getting an increasing number of comparisons for which the data supported a worthwhile statistical analysis. And that shows that the trial design was working. I won't go on to, I don't know why that's suddenly gone blue. It was green just now. We'd go on, and I won't do much on this one, but this one is a stronger test where we start investigating whether there is a trend between the size, both the direction of the differences and the size of differences between plots. Again, we're comparing the farm standards, which the Watkins treated, farm standards with the Watkins untreated, and the same for the Blackman untreated and treated. This is a much stronger test. It requires more degrees of freedom, more data differences than the previous test which we had. And here we had suffered from the same issue of the slug numbers being very, very low. In fact, too low to really support meaningful comparisons in most cases. But the same picture emerges, where we got the ability to make comparisons was always in those sites which had the largest slug numbers. Okay, I'll come back to that in the final conclusion slide, but my penultimate slide now just looks at the slug resistance and wheat crop condition. Now, in these slides, what we have got here is the farmer assessment of crop condition. The solid black line, and on occasions the red one, are the farm standards. So the black one here is the farm standard untreated. The red one here is the farmer standard treated. And where you can't see the farmer standard treated, it was because the data was exactly the same as that contributing to the black line. Key to come out here is, again, we get a very, very consistent difference. In this one, we find that the Watkins wheat is always behind or considered to be a lower condition than the farm standard. Same occurred with the Blackman, only that lasted longer. So Watkins was a consistently lower standard right up until it was well established. The Blackmans remained a lower standard all the way through the trial. And by standard, we think what was happening here is actually we're looking at the rate of growth. And what may have happened is that the Watkins and the Blackman just emerged more slowly and established more slowly in the early stages than the farm standard. So it may simply be a varietal difference. And the same thing occurs here, Watkins below the farm standard, and here Watkins below the farm standard, and here. And in this one, there didn't seem to be any consistent difference. But here, what we are finding now is that the trial is picking up significant differences between the two plots of test plots of Watkins and Blackman and the farm standards. And so a final slide of conclusions. What have we found? Well, the slug sleuths ran an excellent and accurate set of trials or to a standard required for both meaningful conclusions to be drawn and publishable scientific research. And so our primary aim for the first objective has been achieved. Farmers can do really good scientific trials on their own farms without unnecessary intervention from specialists. The surface refuge traps that we caught were found to be convenient. The farmers seemed to like those, and they were efficient for assessing slug activity and risk. We just ran into the issue that last summer was a hot, dry summer, which didn't do the populations any good at all. They didn't recover as quickly as they might have done because there wasn't sufficient rain at the critical period in the autumn. And the result was we were comparing very, very low populations in the resistant wheat plots and the farm standard plots, and the differences weren't sufficiently large to really allow us to draw conclusions. The trap spacing, which we suggested, did indeed prevent mutual interference. And I haven't heard any conclusions from farmers that that was difficult to achieve. And we did get, where we got results, we got consistent results between sites, despite the very low numbers. The trial design therefore worked, and only the low slug numbers prevented a more detailed, stronger analysis and clear conclusions on resistant levels being drawn. But we have confidence that with the standard of delivery and the trial design, that in future years with more slugs, it will work and we can make good tests. Finally, we have some issues now which arise for the year 2 work, which we need to discuss with farmers outside of this meeting and over the next months to make sure we take full advantage of whatever we do starting in the autumn. We hope to have more seed available, and therefore we have to answer questions. We should repeat the basic design in year 2, but for example, do we increase the numbers of traps plots to get more detailed information on slug activity? Do we increase the number of sites? Or do we increase the number of within plot replications? Or do we take a balance of all of this and say we've got a certain amount of work we can do. What is the best way of deploying it? And the photographs, I must finish with this. We were going for pixel analysis this year to see if we could get an accurate assessment of crop coverage at different stages after establishment. But JI, of course, have experts who do this by eye. And we need to conclude whether we actually spend some money developing pixel analysis software, or whether we actually just rely on the good old-fashioned expert eye assessment. So a lot to consider over the next couple or 3 months, but we are really making progress. And I think that we achieved both objectives, and we have A, proof that the trial design basically works, and B, that the farmers delivered it to a really high standard. So Tom, next year is exciting.
Great. No, thank you very much for that, Keith. That's brilliant. And can I just check on one thing before we move on to Simon? Now, I think you said that one of the things about these trials proved, one of the things that these trials proved is that farmers can lay out a trial and follow a monitoring protocol to a level that gives a valid scientific result. Now, is that right? And what's the implication of that? I mean, field trials are an essential element of bringing innovations into the field, and it must be, very expensive in terms of sending research assistants all over the country. So what's the implications, Keith?
The implications, I think you put your finger on it. If we had been doing this trial, then the map you put up in your slides at the beginning shows that we would have had to have spent a lot of technician time traveling between the sites. If they went from Harper Adams, then going up to Yorkshire, 2 to 3 hours traveling in each direction. That means that you have to, if you wanted to do 2 sites up there, then you have an overnight extra expense. The farmers who have participated in this would have taken, in my view, data as accurately as those technicians would have done. And so all that wasted money, if you like, on travel, essential to get there, but on not productive, can go into the productive trial layout and design.
Great.
The other thing, I think, which we need to say, is that I was expecting a little bit of troubleshooting. And we didn't. I don't know if you did, Tom, but I certainly didn't visit any one site. It was all done smoothly. And so the second conclusion is that as long as we have a clear protocol and a really good trial design, then there isn't going to be much need for troubleshooting. And that gets back to what we're going to do over the next months, where we have to talk to the farmers about the trial design, what is feasible, what isn't feasible, what fits in with normal farm practice, so you don't interfere with commercial activities, etc. That's critical. It's not anymore just a matter of a boffin sitting down, drawing up a trial and deciding whether the traps can be 4, 5 or 6 meters apart. There's more to it than that.
Great. No, thank you very much for that, Keith. Keith, do you want to stop sharing your screen? And I'm going to ask Simon now to, Simon, would you like to turn your video on and your audio on and join us? Are you there, Simon?
Sorry, at the moment, Tom, I'm getting you cannot start your video because the host has stopped it.
Right, hang on a second. Ask to start video. There we go.
Brilliant. Start my video.
Brilliant. Sorry for that.
That's okay. I'll try got the presentation. How's that?
Yes, that's great. Thanks.
Great. Okay, thanks a lot. It's a pleasure to speak. So I won't repeat too much that you've already heard from Keith. So this is the Grey Slug, and the work at the John Eyre Centre is very much a joint effort between myself, I'm a wheat geneticist, and Victor Soria Carrasco, who runs the insectary and entomology group And Victor and his predecessors have been very helpful with us in either collecting or rearing grey slugs, and also then doing the screens with us. This is the current team. So this is Mark, Matilda, Will, Anna, and Victor, who I've just mentioned. And I very much encourage you to engage with them in the work that we're doing. So Victor's given a little bit of background here on the grey slug. I don't think we need to dwell on this. And the reason I came to the project was because one of my main drives in the wheat genetics that we do is a wheat landrace collection called the A. Watkins collection. Get my pointer working here. And this is a collection of 827 land races, which means the wheat that people grew before systematic plant breeding began, the wheat that was handed down from through families and between farms in the 10,000 years that we've been growing wheat, over the 10,000 years that we've been growing wheat in agriculture. And this was collected in the 1920s and 30s by Watkins, and 827 is a lot to work on. We used genomics technologies to reduce that to 120, which represents as much as of the genotypic diversity as possible of those 827. And then from those 120, we've developed a lot of other resources that you need for gene discovery. And it means that the 120 is a starting point in many of these experiments. And Jake here, he's no longer with the insectary, but in 2015, Jake very kindly took those 120 Watkins lines, grew them in setups like this. So imbibed wheat seeds on agar plates with a little colony of grey slugs. And you're basically just measuring damage. Jake was measuring the damage to see if we had any slug resistance, because this was a big challenge laid down for us. It was done as part of the DEFRA funded Wheat Genetic Improvement Network. And the kind of the mandate there is to do things that breeders find intractable. You know, nobody's really found much slug resistance in wheat. So we try to do those difficult things that a lot of academics wouldn't want to do because they'd be hard to publish. And in this case, we took on slugs. Now, we were very pleased to see that actually there was some statistically significant variation in the damage to the seeds. And what the slugs do is they come out and they kind of scrape out the seeds, and they generally tend to target the embryos, which of course is the end of the life for the plant, because that's the miniature plant ready to go. And we think this is the most important type of damage. We found some which were particularly tasty for slugs. So these are landrace 704, 127, and 004. And then some possible resistant lines, which wasn't just 7888 at the time, it was also 209 and 325. And our next steps on this work were to take this out into the field. And amazingly, in this case, we defied Keith's rule of, if you want to control slugs or any pest, have an experimental trial on it. In this case, we did okay. This was grown at Morley, which are our kind of partner institute. They farm our land for us, the Morley Agricultural Foundation, and we occasionally grow trials on their land. And we used their land in this case because they grew a lot of oilseed rape. They perform reduced tillage anyway, and they were happy to have a trial with us where we didn't put any metaldehyde slug pellets on at the time, any slug pellets at all. So I remember there was a lot of slug damage. The crop around this trial was very heavily damaged. And in the trial itself, we scored damage and yield on the lines. And you can see that Watkins 788 did experience reduced damage from the slugs. And this was statistically significant. It was analyzed by Lutzi Wingen in my group. So we were really encouraged by this. There was some correlation between this completely unrealistic agar plate situation and a very realistic field in Morley with heavy slug pressure. So you've had, I think at this point, you can imagine I kind of hand over to the presentation that Keith gave, because what we wanted to do was validate this result in real farming situations. Remember, these are, I think these were 6-meter plots, 6 meters squared. Some criticism of it was about we didn't understand slug behavior and slug patchiness, etc. Keith was very helpful in providing advice on that, and the trials that Keith just described were should be even more robust than this experiment. And of course, the fact they're on working farms is a bonus. But anyway, this is where we were, and you see this is 2015. Took us quite a while to get traction to do some extra work, and I thank Tom for firing along with some of that. And I thought next we'd talk about where I'm going with it. I think I've said a lot of this stuff before, but I'd tell you a little bit about what we do when we confirm resistance, and it will help us think about the next experiments. And what we want to do is begin to, rather than just saying, well, okay, this landrace is resistant to slugs, actually understand what genes are contributing to that resistance. And this is important because in plant breeding, it's going to be very hard for a breeder to reliably select using the actual trait to actually grow future potential varieties under slug pressure and then select the resistant ones for exactly the same reason we've experienced. If the slug pressure is low, then the selection pressure is low and the breeder can't make good selections. So what we want to give them is molecular markers, and that's what these things are here. These are all, this is a chromosome, and these are the molecular markers along the chromosome. And what the breeder wants is just a few of these markers around the gene of interest, not necessarily knowing the gene of interest, but around it. And it means that when they cross with Watkins 788 with elite varieties, then they can continue to select for the resistance, even in with no pressure, and in the face of other selection, random drift where they could lose this. We actually maintain it in the families throughout pedigree selection, so that the varieties that come out carry the important genes. That's the idea. And I've just given you an example. We don't have this yet for the Watkins 788, but this is a statistical approach, and it means that you associate the phenotype, the trait, with the genotype, which is those molecular markers, and statistically you can see which of them are most associated with the trait. So I'm pleased to say, because of that early work with the core set and wanting to develop specific resources, that 788 has the type of population you need to do that. And that's called a population of recombinant inbred lines. So it means the whole genome of Watkins 788 is broken up into parts and spread out across 77 individuals. And that's the thing you need to do this association. The material has all been grown at JSC. We've got about, I think on average, 500 grams of each recombinant inbred line. There's 77 of them. And all of them have been genotyped with 6,000 of these molecular markers. So these maps that you can see here, they all exist for Watkins 788, and we're ready to go in terms of doing this analysis. We just need the phenotypic data. We will be very soon, Victor is in the process of running this population in the choice tests again. So we might, hopefully quite soon, we'll have the insectary data. But an important question is, how do we go to the next level, which is the field experiments? I don't think we can possibly do 77 times what Keith just described. It doesn't seem, that would be an extremely expensive and complex project. So probably what we'll do, and again, it would be a nice forum for people to criticize this approach, will be to run the insectary screens, to do some mapping, and hopefully we get results like this, but we might not, let's see. But whichever we get, we'd say, okay, let's take, say 10 or 5 of the recombinant inbred lines, which are very highly resistant, and 5 which are highly susceptible, and then run them in an equivalent experiment to what's just been described. And that should be a sufficient sample to validate Victor's analysis in the insectary. So that's the plan. The approach we're talking about, just for your interest, it's called quantitative trait locus analysis, QTL analysis. And it's called that because quantitative traits are traits which are controlled by several genes, and this may well be. And trait because it's a trait, and locus because these things in genetics, they're called plural loci, and a single one is a locus. So we're trying to associate these quantitative traits to the single loci. Okay, I think that's all I've got.
Great, thanks a lot, Simon. Thank you very much for that. That was really, really interesting. And I just want to pick up on the aspects of the feeding trials there. Now, you picked up some really interesting points there about, you know, what we should be testing. And actually, you know, this is something for everyone who's attending to think about, you know, in terms of where we're going to prioritize some of this work going forward. It takes quite a long time to multiply up some of these these lines, and so the recombinant inbred lines, we'd like to test those in the field, but it'd be good to know which ones we're going to test. So it would be really good to, you know, if you've got any thoughts about this, you know, how we're going to go ahead with this, what we should prioritize, then please pop them in the chat, bring them in, put them in the Q&A. And so on. Those thoughts, even if we don't come to them today, we will capture those, and they will help shape future trials. Now, I was just going to quickly, following on from what Simon said, tell you about being a slug scout. So you may have seen that we're appealing for slug scouts who can collect slugs and send them in to John Innes Centre for to help with this really valuable work that Simon's described. And so, you know, so please do join. Either send me a note or fill in the form on the website, and you'll get a slug scouting pack. So in your pack, there'll be everything you'll need to set up your slimery, and you can use all sorts of things for a slimery. And I mean, you know, I've actually repurposed the door that was fly tipped on the farm, but a piece of rubber matting and things like that would do well. And in the pack, there's some chicken layers mash to get your slimery underway. And we provide cartons to put the slugs in when you're ready to send in a batch. Then there are postage paid envelopes. Now we've checked, and that slugs are live creatures that you're allowed to send through the post. So there's no to worry about live animal transport or anything like that. It's all good. And you'll also get a boffin badge to show everyone the great service that you're doing for science. And you get a boffin pen to capture your moments of brilliance. So anyway, but look, time's cracking on, so let's move on to the slug sleuths themselves. Richard Cross is manager of the Oxton estate in Nottinghamshire. Graham Potter farms near Thirsk in North Yorkshire. Can I ask you both to turn on your cameras and join the discussion? So Richard, I'll start with you. Would you like to just give us a little bit of background about the Oxton estate and why you're particularly interested in this trial?
Yeah, good morning, Tom. Good morning, everybody. Yeah, my name's Richard Cross. I manage Oxton estate in Nottinghamshire. Family have owned this bit of Nottinghamshire for over 500 years. The estate currently is 2,700 acres, of which 1,800 is cropping. We run about 900 acres of winter wheat, 300 acres of oilseed rape, 300 acres of winter barley, and 300 acres of maize or veg on contract. The rest of the estate is made up of woodland and grassland. Two soil types, a red clay, what's locally known as a Kuiper marl, and Nottinghamshire sand and gravel. So we're not interested in slugs on that. You don't get a slug on that, even if you go and sow them. But the Kuiper marl is prone to slugs. I've done bits and bobs of work with Tom over the years when he was a good journalist with the various magazines, and boffin seemed like a good thing to follow, and I've always keen on doing trials work, do bits and bobs with Bayer and one thing and another, because it's always a learning process, and the more we learn, perhaps eventually more money we will make. And we are getting a reduction in chemicals, no matter what anybody says. They're getting taken away from us. I mean, metaldehyde's gone, we've got ferric phosphate. As long as the likes of Severn Trent don't start finding it in the water, we might keep that for a while. But eventually, we might almost be forced down into an organic farming route. So doing this, being at the forefront of this, I mean, we all should be doing it. Helping the likes of you out. You are brilliant as a coordinator. I would do it without the funding, but the funding's nice. So that's where we are, and that's where we're going. And yeah.
No, they're fantastic. Graham, can I bring you in at this point? Now, I know that you've been doing quite a bit of trial work in your neck of the woods. Do you want to sort of tell us a little bit about the farm and what you've been doing?
Yeah, so we're up in North Yorkshire. We have 200 hectares, arable farmer, and also we do a bit of contracting as well. We're on the Claydon system. We have been on it for about 10 years now, been very successful with it. But slugs have been one of my biggest challenges at times with the Claydon. So back in 2019, I took it on myself to do my own slug trials to see how I could try and get rid of these little pesky buggers. I'll manage them a lot better. So I just decided we did a big trial in actually the field that we're doing the trial in at the moment. It seems to be pretty prone to slugs, is that one. So we did an exercise with rolling, double rolling, missing rolling. We looked at different seed dressings, and also we looked at using gypsum as well for controlling them, which worked quite well, recycled gypsum. And also we were looking at different types of slug pellets to use, because obviously we're not allowed to use metaldehyde anymore, so we were using the ferric phosphate, which seems to work very well actually. So that was me trying to understand what slugs did, and also on different soil types. So on our farm we range from blow-away sand to heavy clay, and in our fields they can, it varies such a lot. So I came up with the idea of doing variable rate slug pelleting. So we look at our soil maps that we had done, our conductivity maps, and from there, and also with our yield maps, we can then produce a spreading file for the spreader, and then off we go. So instead of blanket rating slug pellets, we're doing variable rate to the different soil types. And I also I know which soil types are the worst. So on those parts we can put a really good dose on, and other parts we can put a lot less on, or zero. So we're saving on our slug pellets, but we're treating where we need to treat. What else have we done?
Can I ask you, Graham, this trial in particular, what have you found? I mean, was it easy to do the work involved? I mean, for me, it was quite a faff to actually establish the plots, but it wasn't something that we couldn't actually do. I wouldn't say that my plots are the best established plots. So you've seen there.
My plots look amazing.
They do indeed.
I think we need a forage harvester in one of them.
We've got a WhatsApp group, haven't we? And we've been sharing the plot design.
Now the plots themselves. I mean, when I did my trial, that was really easy to do. I mean, I just threw some wheat in straight after we'd finished harvest. That's how I then initially did my trial. So as soon as we'd finished cutting the oilseed rape, I just drilled a bit of wheat in the field, and that's how we did our first trial. And that worked really, really well. This trial that we set up with the slug sluice, it was dead easy. We had it done within half an hour, I think, an hour. With the Claydon, me and my nephew did it, and it wasn't a hard thing to do at all. Yeah.
So Richard, if I can turn to you, I don't know how much of these sorts of trials you've done in the past. Was this your first trial? Did you find it a bit daunting to do it? How did you find it?
For myself, it was a little bit daunting to start with, but I'm very lucky. My main operator has come out of trials work, so it's something he was used to setting up, right. And so he went and set up the trials with the other tractor driver, probably we had the three weeks to do, and it probably took them half a morning, and it was done. And then I went and did the pelleting and the rolling after they'd set them up. So it's not too bad. The monitoring, we didn't go down the app line. We did paper monitoring, but next year we shall use the app, because we've talked about that the other week about how the app's going to develop, and I think using an app to monitor it will be certainly easier, and we'll get better quality data. We've been able to introduce the pictures directly into the app than how we were doing it this year. But it wasn't bad at all. But it's main trials I've done previously have been fungicide trials, and basically that's combine data. A bit of spraying and combine data, which is never really that difficult to do.
Yeah, no, I must say, certainly as far as I was concerned, the app really helped. This is an app that's been developed by Husk Data, who's James Hart, a Cotswold farmer, who's behind that. They've the great thing is they've changed it for us as well. But I'm conscious of time, but I did want to ask you, Richard, about because I know that you set up a slimery as well. Have you seen any slugs yet?
Well, yeah. I followed your line and found a scrap door. Put it probably in one of the wettest bits of one of our fields that's quite close to a pond, so there's plenty of moisture. Unfortunately, the slugs are not enjoying it. They haven't arrived yet.
Oh, well, hopefully they'll.
I did look this morning when we had 4 inches of snow for about 3 days last week, which obviously I didn't want to go out in, so no self-respecting slug would either. But hopefully, next few weeks we'll get some and get them off to Norwich.
No, well, as I say, we are really, really keen for people to get involved with this and become slug scouts. You don't have to be a farmer, but we are looking for grey field slugs. Now, I did promise I know we're running out of time, and I did promise I was going to flag up some of the research work that we've got coming up in slugs for you to take the opportunities to get involved in. So let me just share my screen quickly. So I hope you can see my screen there. Wait a minute, let me just start the slideshow. How can we shape what's yet to come? And we've got first of all we've got funding for there we go. Yeah, we got funding for a second year of the trials, and the project gets underway next month. That includes feeding trials that will continue at John Innes Centre, and we have further on-farm trials too. And hopefully the slugs will turn up this time, and we'll keep you informed of progress. And of course, we want you to help shape it, and this is really, really important. So please do feed in your thoughts, your feedback, and so on. It does really make a difference. Now, there's another project that I'd like to tell you about, but before I do, it's time for a quick poll, if I can get the technology to work. How would you like the slug management to evolve? Given if all of these options cost us, if all of these options cost the same or less than what you're doing at the moment, then what would be your priority as to what you'd like to see come out into the field? And if I can bear with me, I'm going to start the poll. I hope. Hopefully that has come up on your screen. How would you like slug management to evolve? And here are the various options. Would you like to go for patch precision treatment guided by maps? Spot treatment with nematodes using robots. This is really interesting tech, I can tell you. It's been tested in the field. Slug-resistant wheat, well, I think we know all about slug-resistant wheat. Or none of the above. Maybe this is all just pie in the sky. Pellets are all we need, and so let's just carry on with them. Let's see where we. There's. I can tell you that there's a lot of support for slug-resistant wheat, as you can probably imagine, because that's the nature of this webinar. I'm going to end the poll there. Hopefully we can we can share the results. Yes, there we go. So 75% of you are in favor of slug-resistant wheat. Next comes this patch precision treatment guided by maps. Then we've got the spot treatment with nematodes using robots. And I'm very glad to see that none of you have gone for none of the above. So that's great. Now, the reason I'm asking that, there is a reason for it, is that we have a project. And I can tell you that all of those things are going to be looked at in this project. This is really, really exciting. So Slimers brings together all of these innovative approaches in a concerted bid to move away from our current reliance on pellets. Now, within this, there is a sizable budget for on-farm trials, and there's also a lot of testing we need to do on farm. So if you'd like to become a slug sleuth and do some of this work and be paid for it, look out for details later this year. Now, I'm really, really sorry. We've nearly come to the end. I think we've got time for probably just one quick question. And I really do apologize that we've run over on this. But what I'd say is do keep your questions coming in. Send them through to me. My email address is coming up at the end of this session. So we do have one. Let's see if we've got a question. So Kenneth Andrew, if I can, Kenneth, I'd very much like to bring you into the thing. Hang on a second. Let's see if I can find you, Kenneth. Would you like to come in, Kenneth, and ask your question? Kenneth, can you
hear me?
Yes, we can hear you. Yep.
I don't know why he's called me Kenneth. This is Andy Evans here from
Andy.
Chap. So yeah, don't know yet. Yeah, I asked that question. I see Simon's typed in that he's going to look at sugars. It's just that
oh, sorry, could you repeat the question, please, Andy?
Yeah, okay, yeah. Basically, I did some work about 20 years ago where we looked at slug resistance in wheat and found, with the varieties that were around then, and the more tolerant varieties tended to have less sugar in the seed and were leaching out less sugar when the seed started to germinate. And I was just wondering whether that might be a mechanism behind the resistance, in the slug-resistant wheat. And Simon has actually texted or typed saying that he's going to be looking at sugars when they're trying to look to see what the mechanism is.
Simon, do you want to come in here?
Yeah, thanks, Andy. We're actually doing that with Jane Ward, who I'm sure you know. So at the at the Widgeon meeting, Peter Shuri, I'm pretty sure you've made this comment before.
Probably.
Yeah, I think you did. And we are following up on it, and we're doing full metabolomic analysis on the 788 seed. Yeah.
Great.
Hopefully we'll update on that in future meetings.
We've got a question from Tom Dewars as well. I know we're going to run slightly over, but bear with us because it is so important to get the Tom. I'm just going to find you and unmute you. Bear with us a second, if I can find you. Probably helped if I looked in the right list. Seem to have disappeared. Sorry, let me. Tom, yeah. Apologies, I can't find you anywhere. Sorry, we're going to have to move on, unfortunately. Well, look, your question was, can Keith's screen go? Oh, sorry, actually, that was a technical question anyway. Looks as though Tom might have dropped off anyway. So anyway, I'm going to draw the session to a close. Now, thank you very much for your contributions, everybody. If you please keep your views coming in. As you can see, we've got an awful lot of work on slugs, and we do need the feedback from yourselves to help us shape these very, very important trials. I'm just going to bring up my screen. So they do go on to Trinity Global Farm Pioneers, and the web address is just there in the corner. And this is where we can share those ideas. We can put information up about the trials and so on there as well. And it's a place to keep it all together. So please do go on to Trinity to have a look at that. And keep your views coming in to me as well at Boffin. We really do need, your input and your discussion. As I say, when Slimers comes on board, we are going to be looking for more slug slews to take part in trials and make the science happen. Really. So listen, there's my details. Thank you all so much for attending today's webinar. I will be posting the a video recording of it so that you can have a look again. There was some really, really interesting data there from Keith and from Simon to get through, really interesting views. Thank you very much to Richard and to Graham for joining. And thank you to you all and have a good day. So goodbye.